Now my favorite part is doing the infiltration. Again, with the same ring that we use, you can use that for infiltration. You take it, pound it into the ground, and then use some saran wrap and place it over the ring. Why do I do that? So that I can time it and someone can help me pull it and then start the clock and find out how much of that 1 inch infiltrates over a given period of time. Okay? Also, if you pour the water, you may have preferential flow or you may disturb the surface. So when you pull the saran wrap slowly, the water just starts infiltrating immediately. So much about infiltration. So we've covered all the things that we can possibly do with this kit as far as physical properties are concerned. My favorite part is the chemical part. I did soil chemistry, so I like to work with fertilizers and all that. And so pH is one of the things we can do. To measure pH, like Dr. Durant mentioned, you had a meter, and this pH meter and also the EC meter, which measures the salt content, are the two most expensive components of the kit. So to measure your pH, first of all, you take the meter out, and you have to calibrate it, make sure that the pH meter is functioning properly. And to do that, we have standards. We have a pH of about 7, a standard of 7. If your soils have a high pH, we have a pH of 10 for a standard. And for those who have acid soils, like in the tropics, you have a standard of 4. So you do either a single calibration or you do a double calibration, and you go back and check it and see if it actually measures the number out here. If it does, it means the pH meter is working properly. Only then can you go back and measure the pH of your soil. But how do you measure the pH of your soil? You take a scoop of soil, it doesn't matter what volume it is, and you also use the same scoop of water. So if I use a coffee cup and I fill it with soil, I also fill a coffee cup of water and mix them thoroughly. And then I'm going to use that suspension, place my pH meter in that suspension, and press the button and get a reading. Okay? And that reading becomes my pH for a 1-to-1 mixture of soil and water. Okay? Now, with that same solution, you can measure your electrical conductivity. For those who live in the West and those in coastal areas, salt problems may occur. And I believe even here in Missouri, even here in Nebraska, we have salt problems in some areas, right, Mike? Yeah. So with that same 1-to-1 solution, 1 part of soil, 1 part of water, you measure your electrical conductivity. So if you have a salt problem, that would be able to pick that up, and you take appropriate measures to do that. Also, that salt meter needs to be calibrated. So you've got a standard calcium chloride solution that is used to calibrate your pH meter to make sure, your salt meter, to make sure that it's working correctly. So the other thing we can do in terms of chemical analysis on this kit is to measure whether you have a nitrate problem. We know that nitrate is a problem in most of our waters, right? So you take a strip and put it in that 1-to-1 solution, and the color would change. And if you have some nitrate in there, it should match one of these. So if you got 20 parts per million, if it is this pink, or 50 parts per million, now that's when it seems like it is really smoking out there. Okay. So for your chemicals, we have the EC, we have the pH, and we have the nitrate measurement. Okay? Now, the one that has been neglected over the years is the biological properties of the soil. We mentioned earlier that a living soil should respire. But what else? Someone also mentioned that we had about 9 billion microbes in a heaping spoonful of soil. Okay. So how do we know that they're there? It is difficult to count them, because looking at a spoon, can you really see anything? Quite often not. But this morning I would tell you, when I went to collect this sample, we found a toad in there. Okay, we found some grubs in there. So that really impressed me. So I felt like this soil was alive. So what I did this morning was go to the garden out. I don't know if I'm supposed to do that, but I went and dug some soil and put it in here and let it incubate for a while. So what I'm doing is I took one of these cylinders again, trying to make sure that we save on material. And it has some septa in there, so I can use my needle and pierce it when I'm ready to measure, and cap it on the top, and then pound it into the soil. Well, pound it into the soil before I cap it anyway. And what I did next was to put a needle on one side and another needle out here. This needle is going to serve as a vent to help some air movement in there. This has been sitting here since I walked into this room this morning. But you're supposed to measure it for about 30 minutes and then prorate it for the days or year in terms of how much carbon dioxide is produced over a unit period of time. But what would be the problem with that? The problem is that you're capturing the carbon dioxide maybe in the morning. You know, respiration varies with what? Temperature. So the warmer the soil, the more respiration is taking place. So those averages are not very good sometimes because it depends on how hot it is. Is it in the winter that you took your measurement? Was it after a rain? And those kind of things. So you must make sure you take care of all that. That is part of the interpretation. So I need some volunteers. Who's got some muscle to come and pull this up and see if we can figure out how much carbon dioxide has accumulated in this chamber over about an hour period? Anybody? All right. Okay. While he's doing that, I'll pass some of this around. So we have a syringe that is about 140 cc's, but we pull it only to 100. If we pull it the first time and it gets all blue or it changes to violet, then that's fine. We can stop after one shot. But if it doesn't, we may have to pull 5 times. So we'll be sampling 500 volumes, 500 cc's or 5 volumes of 100 mils. Okay, you ready?
Yep.
All right. So you pull till it gets here, you take it out, press it so it doesn't leak, and you start again. All right. Okay, thank you.
Ready?
Yeah, keep going. Oh, that's all is smoking indeed. Huh? Can you see that?
Hold it there now.
Okay. I did. Pull it out. Pull it out. And then push it all the way back.
Okay.
All right. Then feed it back in. So you need 2 people to do this kind of work sometimes, huh? All right.
100 again?
Yeah, pull it. Pull it to 100. All right. So
All right.
Can you guys see a change in color? Can you notice that? So this is in percent carbon dioxide. Oh, you can go 5 times. Why not? This is getting exciting here. That's all is alive.
You got it?
Yeah, I got it.
Yeah. Okay.
All right. You got it?
Got it.
All right.
Oh, it's like, you know, on there.
All right.
Okay.
What's the little filter thing that's turning green?
It's turning blue. You need to use some eyeglasses out there. Okay, well, it is a carbon dioxide sensor, and once the carbon dioxide hits it, it changes color. So
Was that 4 times?
that was 4. You see, it takes a while. One more, I'll be done. Perfect.
Okay, there
You got it.
5.
All right.
Got
Okay. Thank you very much.
Okay.
All right. So you can all see the color change, okay? And that tells you that, you know, we have about, how many percent do we have there? About 2.5. I need some glasses too. 2.5% carbon dioxide for 500 cc's of sample. Okay? So you divide that by 5 because we did it 5 times, right? To get the volume, to get the percent carbon in 100 cc's. And then we've got a formula in the book. We also got a software that helps you calculate that, and that translates into how much carbon dioxide is coming from that field over a 24-hour period or during the day on the average. Now, we mentioned earlier that the respiration rate depends on the temperature. So we ought to have this. I didn't really figure it would be in the package all the time, but we've got to have that in the soil. Make sure that we know at what temperature this is occurring. So put that out there, and your temperature can be read either in Fahrenheit or it can be read in centigrade. So that's another way you can help your kids do some calculations, convert from Fahrenheit to centigrade. So there are a lot of things you can do out here in terms of math, in terms of history, in terms of interpretations, and related to the land. So I got a little sheet that I passed out. I've also got this package of a folder of handouts. They have our web page on soil quality. They have some of the tests that I just described, and our website is on there if you need more information. We also have a CD in here with lots of lesson plans and tools for educators that you may be interested in. Okay, and there are other resources I believe that Brandy put together with websites that we assembled together to help you prepare your lessons. And the lessons that you present will be those that interest you. Some interest me more than others. I like to do this one because it shows a dramatic change in color. Okay? So, the calculations on bulk density, soil components, moisture content are on the back of this flyer that you have on your desk. Okay? And if you don't remember anything during this presentation, I want you to remember this, that the quality of our life depends on the food we eat, the air we breathe, and the water we drink. And all these depend on the quality of the soil. Thank you.